defines herself as a theoretical statistical physicist with broad interest in understanding collective phenomena, topological defects, and pattern formation in complex dynamical systems. She comes originally from Romania, but she did his master in the Niels Bohr Institute in Copenhagen. After this, he moved to Oslo for the PhD, and she got the PhD in 2010. And after this, he has been collaborating with many people. In particular, she was a visitor at IFISC in 2018, and also a visitor at the Kavli Institute in Santa Barbara in 2020. She works in a lot of topics, from condensed matter topics such as dislocation and plasticity in solids, liquid crystals, active matter, interfacial dynamics such as the interface between ice and rock, and superfluid Bose-Einsteins, the dynamics of Bose-Einstein condensates, and also some other things of biological interest such as tissue growth and tissue morphogenesis. And I think this is the topic of today, tissue dynamics due to topological changes and fluidization. So thank you for being here, and the floor is yours.
Thank you, Emilio, for the kind introduction. It is very nice to be back here. I was here with the fellowship several years ago, about 8 years ago. So it's nice to be back and recognize many of you. Yes, so as Emilio mentioned, I'm based at the University of Oslo. We are part of a condensed matter physics group in the Department of Physics. The research that I carry on is very much rooted in statistical physics of complex systems, and I'm interested in system with in ordered systems. So there is an underlying topological order, and then they acquire topological defects, and there's all kinds of systems that I have explored over the years. Started with the superfluids in this context, and we've worked on quantum turbulence. There's three main directions. So this is one direction that also Emilia mentioned. And this is quite interesting recent work that is done in collaboration with the group of Mahesh Bandi at Okinawa Institute of Science and Technology. And here you have experiments of draining water tub. So there's a container with the sink, and then there's a vortex that is formed there, just the one that you see in your bathtub. But then there is a constant injection of fluids, of water, so that there is the height is constant. And then you introduce shallow water waves. So there are waves coming from the left and from the right, and then there is this spin vortex there. So we're looking at the interactions between vortex and waves in this context. And what is interesting here that you see the analog of the Aharonov-Bohm effect. So there is a phase shift, like the Berry phase, that is acquired by the incoming waves. This has been known before for a single traveling wave. And there is a disclination that is formed in the stripe pattern. But what's interesting right now is that we discovered that there is a whole line of singularities if you have standing waves, and these are what we call the nodal lines. If you look at the phase of this periodic profile, you see the discontinuity line, and this phase is rotating, and this is something that we're currently working on. And a similar example you see if you have a single vortex in a Bose-Einstein condensate, you pin it. Well, you don't pin it there, but it's just stationary, and then you shoot the shock waves. You see that the shock wave basically splits into two transient phase slips, and one of the phase slips annihilates with the actual vortex, and the other phase slip becomes the new vortex. Okay, so it's an interesting phenomena, but that's not something that I'm talking about today, but I will be happy to delve into it if it sparks your interest. Another line of research has to do, is more at the interface with material science, and there we characterize topological defects in periodic patterns. So we have a crystal lattice, and I should mention that we are using, there's all kinds of methodologies. We're using a mesoscale or a field theoretical approach. So we have a density field that gives you this periodic profile. And from that, we can extract the dislocations by looking at the phase of the magnitudes associated to each of these periodic modes. And we could do this also for real crystals, but also for quasi-crystals. And that's a very interesting work that is done in collaboration with Marcos Avaloglio's group at the Technical University in Dresden, and also with Jorge Vinals from the University of Minnesota. The other topic that I picked up rather more recently in the last 5 years is the topic of active matter, where you have particles that, as we heard yesterday at the PhD disputation, there are particles that have this intrinsic motion because they consume energy from the environment and they turn it into translational motion, and that drives the system out of equilibrium at the level of the individual particles. So the detail balance is broken down, the time symmetry is broken down at the individual level, and that poses very interesting conceptual and challenging questions to understand what happens with the collective behavior. So I work on this interplay between understanding the feedback between topology, geometry, and flows when you have this active ingredients, and that's the topic that I will delve a little bit more today, specialized in particular for understanding tissues. So let's start from sort of somewhere, not the very beginning, but from somewhere. And the question would be, how do cells move when you have a tissue? Think about the skin, the cells in your skin. How do they move? So we looked at what the biologists are telling us, and essentially there are two very different modes of locomotion. It's one type of migration, what's called the mesenchymal migration, is basically the type of crawling. So imagine that this is the cell. So we're talking about eukaryotic cells, so the cells that have a nuclei, so not necessarily bacteria and other kinds of colloidal type of particles. These are actually eukaryotic cells, so they have a nuclei, and they are on a substrate. And they do this crawling motion, so they have this lamellopodia, this fingers that are attached to the surface, and one at the rear end detaches while the one at the front attaches. So you have this crawling motion. And when you look from above, you basically see that the cell stretches in one direction and then contracts in the other direction. And it stretches preferentially in one direction, but there's all kinds of fingers. So it explores the space with its fingers, and whichever finger attaches, then the other one detaches. And they can cluster together, but the contact between them is rather loose, and they very easily can migrate away from each other. So they can be together in a pack, but they don't have to. And cancer cells like this type of motion. Cells in the muscle, the fiber cell, are also elongated, and they also like this kind of motion. By contrast, there is also what's called the epithelial migration that is very much a collective type of mode of motion, in the sense that the cells are strongly bonded with each other. So they have what they call stable cell-cell adhesion bonds, and they like to stay together, and they prefer to be in a dormant state, and then they can get activated. And when they get activated, they are very much coordinating their motion depending on what the surroundings are doing. So you have this coordinated motion, which might look like, okay, we know synchronization in physics, right? We have this flocking behavior. So indeed, they can move together as a flock, but that's not very efficient, you know, to have the whole cells moving. There can also be a lot of localized migration relative to each other. So that's what I call the fluidization. And the way they move relative to each other in this kind of hexagonal packing, it's something that is not fully understood. What are the physical mechanisms? And you know, if you talk to a biologist, they might say that there are all kinds of biochemical cues that are guiding this migration. As physicists, we also see that there is a lot of physical instabilities and mechanism that drives this. So there's an interplay between the two of them. One characteristical difference between these two is the fact that when you have an epithelia, like the tissue, the lining, so you have this compact lining of the internal organs, but also on the outer skin. It's the largest type of epithelia in the body. The cells that are making this epithelial, they have two types of primary polarization. One is called the apical basal, so top-bottom polarization. So when you have a surface, it basically generates the orientation of the surface. The AB polarity is associated from a physics point of view with the normal of the surface. It's perpendicular to the surface, right? And then you have also an in-surface polarization, which is called the planar cell polarity, and it's this asymmetrical distribution of some some proteins, these PCP proteins. So this is marked here in red and in blue, and these proteins they have this they polarize, they concentrate at certain junctions that are in the opposite end. Okay? And this polarization of the PCP protein generates an in-plane polarity. And what we'd like to understand is, what's the role of this polarity that makes this type of motion very different than that type of motion. This type of motion has been analyzed before in the context. Turns out that it's relatively well modeled by colloidal particle physics, liquid crystals type of thing. Models from the liquid crystal physics match very nicely this type of motion. But there's currently no, unified model that describes all the complexities of a epithelial solid. And there's all kinds of initiatives right now. It's a very active field in the topic of active solid, because it has a sort of solid nature to it. So what kind of model that is, that's something to discuss. But of course, in reality, what I've showed you is the ends of a spectrum. So the cells actually can undergo a spectrum of migration. And in fact, in cell biology literature, there is also what they call the mesenchymal to epithelial transition, or epithelial to mesenchymal transition. So the same type of cell can change its mode of motion from one to the other. And this transition has been associated to metastasis, cells kind of becoming cancerous when they undergo this transition. And there's very nice work done by colleagues from the active matter physics trying to understand the nature of this phase transition, from the physics of phase transition that we know. So what I would like to pose to you today, something that we've been asking ourselves and trying to dig a little bit more into the model, like what is the effect of this type of polarities, the planar cell and the AB polarities, in destabilizing this stable cell-cell contacts and triggering this localization? What can we make a minimal model of that? Just to kind of give you a little more context why this is important. It's important for all of these things, for the development. So you want to have rigidity of a solid packing, but you also want to have the fluidity for morphogenesis, to change shape. And it's very important for metastasis, for regulating the functionality of the tissue. When tissue goes cancers, then they become fluidized. And it's also very important for tissue repair. If you have a wound, like, you want the cells to be able to very quickly access the wound and repair it without disturbing the whole tissue. So, okay. So we'd like to understand a little bit more how do cells transition from this solid-like to fluid-like response. And because of the nature of this close packing, there must be some underlying order in these polarities. And if that's so, then what is the effect of distortions, topological defects, in driving this polarization? So with further ado, let's say that now we can go into the main outlook of the talk. So I'm going to present to you two different types of models. Particle models, but very, very different in some way. One is the particle model in which the cells are idealized as point particles, but they have this extra degrees of freedoms, which are these two polarities. So think of an XY model, but now we have another polarity, another spin vector. So we have two spin vectors, and then the spins are actually mobile in space. So there is also the velocity. And the way they move is dictated by the relative orientation of these polarity vectors. And we like to understand in this minimal model what's the effect of the planar cell polarity on migration and the fluidization. And the other type of model assumes that we don't care about the polarizations, but we track the cell boundaries. That's very important in a close packing. Who is the neighbor of whom? And we track the cell boundary as a diffuse interface. So the natural model for that would be a phase field that is 1 in the bulk of the cell, and then it very quickly transitions to 0 outside of the cell. And in this formalism, we have as many phase fields as cells. So computationally, it becomes very expensive, so we can't run thousands and thousands of cells. So the order of magnitude here is of the order of a couple of hundreds, maximum 200 cells. That would mean having 200 phase fields coupled to each other. And then there are some interaction terms between these phase fields that tells us what happened at the junction. This model is very suitable for studying growth, division of the cells. And it's also very suitable for understanding junctional remodeling. What happens with when they exchange neighbors? Essentially, a junction disappears this junction would disappear and another junction would form. So you have this cell shape changes and junction remodeling, and what does it do to the collective migration. Okay, that's the platform. So let's start from the discrete particle model, the spin model. We start from a phenomenological interaction potential. This is purely phenomenological. And it's a bit informed, sort of, by intuition and what kind of works. So we would like to have pairwise interactions, particle I and particle J. Each particle has these two different polarities, call them Ni for the AB polarity and PCP for the planar cell. And then we have the separation distance vector between them. So we have an interaction potential that basically has three terms for all kinds of three types of interactions that we may have. And by construction, this interaction potential is anisotropic in the sense that it depends not only on the magnitude of the separation vector, but also on the orientation, and that is through coupling with the polarities. And essentially, we would like a ground state in which all of these polarities are perpendicular to each other. The separation vector and the PCP are two vectors perpendicular to each other. They define the plane. And then the norm to the plane, the normal to the plane, that's the AB polarity. So based on that sort of geometric construction, we sort of have this penalty terms that this is basically 1 when these vectors are perpendicular to each other, and then it may not be 1 depending on the different orientations. Okay, so based on this interaction types, we can then write an overdamped Langevin equation with the gradient forces acting on the particle migration, and we have some translational noise. And for the polarities, we say that they are of unit norm for simplicity. And there is only rotation, so there are torques. So there are these torques that are induced by the corresponding gradients with respect to each polarities. And then we also have the rotational torques, this rotational diffusion to each of them. Okay? And notice that this kind of torques depend only on, so if you look at the PCP polarity, this P vector, it enters into these two S terms. And one of them does not depend on the separation vector. So we call more like this handiness. So this type of torque rotates the PCP vector so that it aligns perpendicular to the AB polarities, regardless of the separation between them. So that kind of gives you a handiness. Whereas the other one is the torque induced by the separation between the two particles. That sort of plays a role in the later on. Yes, this is in 3 dimensions. Yes. And now, how do we fluidize? The fluidization is a geometric rule here. So we start with a connectivity matrix in the beginning. Okay, they are point particles. So each particle has some neighbors. And then we define this rule that if the distance between two neighbors, we consider that they are closest to each other, remains an active distance. So A is a neighbor of B as long as D is far away. But if D comes closer, then we say that A is not a neighbor of B, but C becomes a neighbor of B. So we have this sort of geometric rule so that we have nearest neighbors interactions. Each particle interacts only with its nearest neighbor defined by this connectivity graph. And this connectivity graph changes in time because of this geometric rule. Okay? So this is a short simulation of very few particles, just to kind of get an idea of what happens when you have two particles. This, of course, a point particle, so we make them like spherical. You can also add a short range repulsion for numerical stability, but that's not something that we explored consequently.
2D or 3D? That looks 3D, no?
It is 3D.
3D.
Everything is 3D.
Okay.
What about the interaction potential depending on the distance? Is attractive, repulsive, mixed?
It depends. It depends on the orientations. If two particles have orientations that are, if the polarities are aligned in the same direction, they will attract. If they don't, then they will repel. It depends on the sign of S.
There is no specific interaction with the distance. Except this short range repulsion.
With the distance, yeah, in the sense, there's two terms here. But it's not like an elastic spring. Although in 2-dimension,
That depends on the S.
It depends only on the vector, not the distance. There is no other isotropic contribution. We could add an isotropic contribution to that. So this is a very minimal model that you can add ingredients. Actually, in 3 dimensions, when we go beyond surfaces and look at aggregates. Very nice question. Thank you for asking. That is the unit vector. So it does not depend on the distance. That was not depend on this. Sorry. Yes. Yes. It's a purely angular dependence, if you want. Yeah? It's an angular dependence here. No, we could add it. We could add it, but and numerically we did add it. But I'm going to do a coarse graining of this model, and in that case, we just consider the unit separation vector. Yes, correct. Yeah. And so that was the simulation that shows you that they will align. In this case, when you have 4 particles, they will move around, and eventually they align to a configuration when they form a string, so that you have the perfect orthogonality condition between these vectors. But of course, we want to go to larger scale and many systems. So now I have a small excursion into topological defects. I can very well skip this slide if you are familiar with the topic. We'll go ahead. So these topological defects, by definition, they are high energy excitations from an order state. I always have a problem with this slide because it just runs away on its own for some reason. There you go. There you go. The easiest way to imagine it is that you have a vortex. Okay, so there's this circulatory motion around it, and they induce, even though they're very localized, the defect is located here, but they induce a pattern that is very long range. In fact, it's something that cannot be coarse-grained, so there is no cutoff scale. You would feel those defects even on the edges. Okay, so therefore, these are very localized distortions that have global effects on the scale. They cannot be considered in the same class as small scale details. You know, in physics, we have the small scale details that they average out or they give effective effect. They are at the small scale, but they have global effects. So let's suppose that we have this pattern. And let me define you the topological nature of this vortex, why we call it topological. Because if you look at the unit vectors, and you now take the orientation of the vectors around a closed loop, so this edge is green, that one is blue and orange and yellow. So now I take all of those vectors and I put them on the circle. So I deform. This is called an affine deformation. I have a smooth deformation and I deform the square into a circle. All right, now I have all of these orientations along these edges. Okay, now I do another affine transformation. I translate all the vectors so that they point in the center. They have the same they have the same origin. So now these unit vectors are pointing around the unit circle. So we see that as I do a loop in the real space, in the parameter space, basically the vectors are tracing a path on the circle. So the circle is basically the values. Each point corresponds to a values of the orientation, and a loop corresponds to a whole circumference travel along the unit circle. Now, topologically, I cannot shrink this loop to 0 without leaving the unit circle. Okay, that's called a mathematical singularity, because this is any type of deformation that I've done here by deforming this would be a type of small path on the circle that can be shrunk. But here, because I go all around, I cannot shrink it without leaving the domain. So it's a singularity. The way we can represent it more like as a physicist is to construct an orientation map. So from each point in space, it stresses me out, this thing. Yes, from each point in space, I can construct the local orientation vector, the local orientation, okay? And I can plot that orientation field, and then you see that there is a singularity, and there is the branch cut associated with it. The branch cut is completely arbitrary, right? That's like in complex analysis, but this is the singular point. Because if we do a loop integral around it, which corresponds to going around the path, we get a phase slip. We do not come back to 0. And the phase slip is quantized in units of 2 pi, and that quantum is the winding number, okay? And the winding number is typically an integer. They can be also fractional, but in this case, for the vortex, it's an integer. It could be +1 or -1. They could also be multiples of, it could be of order 2 or 3. They are called higher order defects, and energetically, they typically are unstable, and they dissociate to the elementary one. So if you want to have a zoo of the elementary defects associated to what we call polar defects, like in the XY model, you have this vectors. So it's an orientational and directional order. Then we have, for m equal to 1, we have essentially in the class of vortex. Vortex is one type, but we have slightly different patterns that have the same topological charge, but spatially they are very different. And the spatial flow is dictated by the orientation of the far field. You know, the far field, it acts like an intrinsic angle that gives you the pattern of a vortex, spiral, or aster. The negative one does not depend on the far field, which is this one, if you want to represent it like here. This is the constant phase. That's going to give you the direction of the vectors, the uniform direction of the vectors. Okay? And this is the phase relative to the position of the vortex, which in this case is at the origin, and this is the m ± 1. So this is how we represent the polarization associated to a vortex. Okay, so now we go to the numerics, and we run that particle model where we seed a vortex or an aster, a vortex, an aster, a spiral, or a negative defect. We keep it there, which means that first of all, we are in the plane, so the AB polarity is out, it's constant, doesn't affect. The PCP polarity is imprinted. I have this pattern that I keep it there. So the only thing that actually is in this code is to run the Langevin equation for the translational motion to read off the velocity field. Okay, and we like to see how much the velocity field reflects the pattern. We have the same pattern or not, okay? And for the aster, yes, the asters can be inwards, where the vectors point inward or outwards, right? We can play with the imprinted aster, and regardless of the PCP aster that is inwards or outwards, the migration is always outside. So cells tend to move away from an aster configuration. And then as you go towards a spiral, you see that the migration starts to kind of move at an angle relative to the PCP pattern. It becomes also a spiral, to the point when we get into a vortex, it actually points perpendicular to it. And it always points inwards. So the lesson that we get from the simulation is that the asters act like repellers, cells do not like to be close to an aster configuration. But if there is a vortex configuration in this cell polarity, they tend to segregate there. They go into. Which means that when they collide, they can actually lead to surface instability potentially. This is kind of a buckling effect, right? And for the negative one, the PCP defect has this quadruple pattern, but the migration has this 8-fold symmetry in here, and it leads to this pillowcase. Of course, it's not consistent with the Euler characteristics of a disk that is +1, so you have a -1. So then the geometry will change so that you have the shape that accommodates the topological charge through the Euler characteristics. Okay, so let me go a little bit more. So we've done a coarse grain. There's a lot of calculation in here, a la Shmolukovsky, basically looking at probability density, and from that getting the evolution of the particle density and the evolution of the polarization. And for the particle density, we get the conservative dynamics. There's nothing there, yeah? With the velocity field that is given by this form. Remember, this velocity fields come from these interaction forces. And if I trace it back here, this interaction forces, when I take the gradient, you see that it's quadratic in P. It comes from here. This one is the AB polarity, which in 2 dimensions disappears. So the only term comes from here. So I take a derivative with respect to r, so I get another r that kind of goes away with the coarse graining, and I have this quadratic thing. In quadratic form in the polarity, it's something that is very good in the mean field. It closes in the mean field very nicely. That's what I'm trying to say. So we get a very nice flow pattern that, for people that are familiar with active pneumatics and active matter, hydrodynamics of active matter, they will see that this is basically like the active stress. Is a force, is a flow essentially induced by dipolar forces. So in other words, these cells act like what's it called, this yes, the cells have a dipole essentially, and they create a flow field around it. In the language of bacteria or like other micro swimmers, that's what I was like, colloidal particles. You have swimmers that swim by pulling by pushing in the air, or by pulling, by swimming against the flow or dragging the flow in. That's basically what the cells are doing. They're either pushing into the other cells, or they're dragging the other cells for them to kind of move. Okay, so they create this dipole, and these dipolar forces are generated by the asymmetric distribution of this PCP proteins across the. So in some way, it's a junctional stress. But the interesting thing is that this is an active stress that appears in this model without actually adding activity. I haven't made the particles self-propelled. If you notice, everything is gradient forces. Right, gradient forces, and still, which in most of the other systems, and this is the question that I've been having, and this is work that it's still in progress, so it's not published, so it's still stuff. Like, where do you break the detail balance? That's stuff that I still don't understand, because this active stress is associated with self-propulsion that breaks the detail balance. So how do you get this active stress here? I'm asking this question to myself because this is something that we understand, but it's yeah, very interesting. And of course, we checked this now. If I do the same thing as with the numerics, I imprint a vortex or I imprint a topological defect in the PCP profile, and then I read off the velocity field. I see a very nice agreement with the prediction from the simulations. Okay, so indeed, for a vortex, I always have an incoming flow. Aster is always repelling, and for the negative one, we get this eightfold symmetry. So we know that the coarse graining works. Now, no, that's the thing. It's compressible. Yes, very, very important thing. Now, as you mentioned, this is a 3-dimensional model. We take it off the substrate. We don't want to keep it flat, so we want to see what's the role of the AB polarity. But we start with the same thing, but now we don't constrain the AB polarity to be in the Z direction, so like put the tissue on a substrate. So we start with an aster, and with a little Gaussian bump, it's hard to see it. But when you have an aster, basically the Gaussian bump flattens out. So it's exactly what we are predicting in 2 dimensions, that the cells will tend to go away from an aster. But in this case, you have a vortex, and you see that the cells are, eventually it's going to turn into this. You start with a little bump, and the bump keeps growing. So the cells are accumulating at the tip of the vortex, and it generates a mount, and the mount can develop into something, a morphology. So we took this idea further now, and we say, okay, now we have the vortex, and that generates a curvature. What would be the asymptotic shape that we get? All right, but now fluidization comes into the business. Are we letting the cells exchange neighbors or not? In the case where you don't, then we have this more like solid response, because everybody sticks together and they just have to move in this coordinated way. And when that happens, then consistently we see the formation of a cone. which is very, very nice. And we characterized a little bit the dynamics of this cone. One question that I got at some point is the cone opening. How is that dependent on the model parameter? That's something we still need to do. The cone is empty. Yeah, so you start with the flat surface, put a vortex in the PCP order, and then the cone is formed. Now, what happens if you allow for neighbor exchanges? Something very interesting happened, same initial condition, but now if the cells are too stressed, if there is frustration in the connectivity matrix, they can just leave the neighbors. They can exchange neighbors. When that happens, you see, the cone actually opens up like a volcano, and then it develops a tube instead. And this is a map of the neighbor exchanges per cell. So you see that most of the activity, maybe I'll run this again, most of this neighbor exchanges happens at the tip of the vortex. Okay, you see that, and there are some streaks. The color scheme is not that. We try to understand that there are some fault line that develops far away, but most of the time, these cells here, they move together. There is no frustration in the connectivity matrix, but here the top is where the thing. And so this was very interesting when I showed it to my cell biology colleague, because he was like, "Well, that is absolutely fantastic because we are full of tubes." Tubes are everywhere. So then it's a very minimal model for the spontaneous formation of tubes. Okay, so how tubes, in this complex environment that the cells are living, why are the tubes such a prevailing structure? And we say that it's basically topologically driven. There's not much, lots of other things do not matter. But fluidization matters, that's the thing. You need the right topology and the right fluidization mechanism. So I know that my time is kind of running out. But I want to tell you a little bit more of something that we do now that we're beyond a surface. What I've shown you is a surface on a substrate, off a substrate. Now, what happens when you have an aggregate? When you have an aggregate, we can look at the elongation of this aggregate, which it's elongation. It's a spontaneous symmetry breaking that you start with something that's spherical and then it elongates. And that's a primordial transformation that happens in embryogenesis. You start with the fertilized egg, and then eventually it creates an aggregate of cells. That aggregate is spherical initially, but eventually it spontaneously elongates, and that creates the body axis. So for many of this tissue developmental processes, the initial elongation is the onset of the body axis formation, how the axis on which the body forms. So now we say that that is actually due to this, it's also topologically controlled. The PCP polarity tends to align perpendicular to the main elongation when that happens. If you look at the intrinsic migration, you have this convergent extension flow. So it contracts in one direction and elongates in another direction. This is all nice, but what's very exciting is that we have actually access through our colleagues from the medical life science. We have access to actual gastroloids, from stem cells. They are prepared in a way, and we can track the nuclei. So it's basically the stem cells that are pluripotent. So you have an aggregate of stem cells that are immersed into a biochemical soup there, and they are exposed to all kinds of cues, so they can be trained to differentiate into lung cells or into heart cells. So you start from this stem cell technology to generate differentiated cells. So you can create like an artificial eye or an artificial heart or lung, starting from, and the gastroloids is like the primary thing before they differentiate. Then they become organoids that are a little bit more specialized. And an organoid is like a baby organ. And there's a whole technology in this kind. And right now we have this essentially stem cells that are guided somehow to clump together. And we track their nuclei as a proxy. The nuclei of those cells, they are a little bit elongated. And what we've been learning is that the anisotropy of the nuclear orientation of the cells is the proxy for the PCP polarity. Not exactly, but it's the best that we could do. All right, so we do a principal component analysis to extract these eigenvectors associated to this ellipsoidal kind of nuclei. And from that, we can get all of these polarities, and we can run a systematic data analysis for the kind of collective order in these polarizations and the correlations. And the colors are the different nuclei, essentially. The staining. This is the staining of the nuclei. And you can see that one important thing that we neglected in this model is, of course, is also the volumetric growth. Right now, we're looking at just shear elongation, preserving the number of particles. But in the early stages, you see this Gauss rate is much smaller than that one, or this one, because the growth is very important. They also multiply at the beginning. It's growing in the experiments, yeah. Yes. As a crude approximation, we say that the growth just add the volumetric expansion, and we took that away. But the truth is that it might not be the case. Maybe the growth is also a source of anisotropy. While we learn that the growth is very, very important. Right now, we only looked at the effect of these intrinsic polarities that leads to the elongation, but that would be something to look into it. So this is something that we're still kind of like wrapping it up. It's just as a teaser, I put it there. Now, I know maybe if I have like 5 more minutes, 10. 5 to 10, yeah. I can kind of go a little bit, because that's the part that we kind of do. The other one is like the phase field model. And the phase field model, as I mentioned, has junction between the cells as a way of labeling the neighbors. Here we have a graph with the connectivity between them. Here we have junctions, you know, and the neighbor exchange associated with this formulas when we have junction means that there is a topological transformation in the sense that a junction disappears and another junction is formed. And typically a junction disappears. So this is a junction. No, this is a junction. C and D are together and they have a junction. This junction disappears and it forms a little hole. And then another junction is formed perpendicular to it. So A and B become. So because of this orthogonal type of formation, you shrink a junction this way and you form another one. This is called a T1 transition. Okay, there's a typical type of neighbor exchange that happen in foams and in other type of granular matter, and now it's been used extensively to describe confluent this tissues. Okay, so let's explore the effect of fluidization due to that. So this is a numerical simulation of this phase field model. I'm not going into the detail of the formulation because it's quite heavy. It's kind of a standard model. It has an advection. So in this case, this is an active model. The particles are self-propelled. So this advection is per cell, you know, per cell. And the velocity is the V-check model. So it has a constant magnitude and an orientation that follows a random walk. Yeah, this is
alignment with the neighbors.
No, it's random. It's V-check. It's V-check. No alignment. No alignment. Yeah, that's a very important point. No, sorry. V-check does have alignment. No, it's an active Brownian particle. It's the V from the active Brownian particle. Yes. And you see that, this T1 transition essentially are activated by this self-propulsion. The cells always have this drive to move around, and if they can move together with the neighbor, so they have a cluster that move together. But if not, then they will exchange neighbor, and you can see here we've colored out some of the cells, so you can actually track the migration of just few of the cells. So you see that there's quite a lot of migration, quite a lot of motion in there, and we try to quantify this a little bit. Maybe I'll skip about this just to show that.
There's a lot of migration, but also including some kind of self-propulsion velocity here?
No.
There is no self-propulsion.
Sorry, there is this is only the self-propulsion. This velocity is just the self-propulsion. Yeah, so the cells have a center of mass. So this is a center of mass velocity, but then we extended it to everywhere in the phase field. So the whole phase field is moving with the same thing. Is that clear?
Yeah.
Okay, good. This is just to show that every time when you have this junction remodeling, there's an energy drop. So it's a very dissipative process. I can skip this. This is more interesting, that when we average the flow or the velocity field from the self-propulsion velocity, the center of mass. This is the self-propulsion velocity, coarse-grained, so we have a flow field. around the epicenter of a T1 transition. So we have all of these T1 transitions that have happened, all of this junction remodeling. And then we have a mechanism of rotating them so that the principal axis of annihilation of a junction, the formation of a junction is orthogonal because they can come in very different directions. So we rotate the velocity field so that we have the axis in which you lose the junction is this way, and the action in which you form a junction is this way. Okay, and then we average that velocity, and we get this very nice quadruple flow field to show that essentially they generate vorticity. They generate these persistent flows. Now, this is an event that lasts for a very short time. It's very quick, but this flow, it's quite lasts for a much longer time than the actual time of a T1 transition. So in that sense, it's a persistent type of flow, but eventually it will decay. Okay? So it has this mixing. Another thing that we've done is to track the particles, and we looked at the mean square displacement, yeah, relative to some initial condition, okay, to see how diffusive these particles are. And you see that it's essentially super diffusive. The slope is always larger than 1. And that makes sense because they're self-propelled. But when the activity is very high, it actually gets into the ballistic regime, which is basically following the self-propulsion. They're just moving so much. And in the intermediate range, you also have this scaling between normal diffusion and ballistic regime. There's not much that we learn out of that, but then we looked at the pair separation that has to do with mixing of the cells and how they change neighbors and how far they move around, kind of like the Lagrangian exponent. We didn't go into the Lagrangian exponent because we didn't know if it's hard to see whether this is an exponential separation. The time scale is very short, but yes.
You don't get any kind of, this is a good question. For very large self-propulsion velocity, any kind of Mises phase, there is some kind of phase separation, diluted areas or no?
That's a very good question. That could happen if we have different magnitude, different populations. But in this case, the magnitude is a global quantity, so all the particles are moving fast then. But there is not that. No. There is not that phase separation that you would see in the Vitek model, and I think that's because they move. I understand the phase separation, and I think that's the reason. The reason we don't see that is because this is closely packed, and we have periodic boundary conditions, so there's no way to segregate in space.
Can you describe that? So the cells always touch.
Yeah, they always touch. So there is no.
By exclusion, this is all the part.
There are no holes. Yeah, there are no holes.
There is no possibility.
Yeah.
Separation of.
But it can be done. In this formalist, it can be done, but we just didn't do it. Yeah.
I don't understand how to define frontier. So the shape you show from the phase field, and how that comes out.
That's the phase field profile. So that's like a tangent hyperbolic that interpolates between, for each cell has a, and then this shapes, this tangent hyperbolics, they are overlapping. And there is a potential that penalizes how much overlap you have. So you want a little bit of overlap to create adhesion, but you don't want too much overlap so that you conserve the area and you don't have the cells penetrating into each other. So that's the part where you sort of tweak a little bit more with the potential fields and
Cells close by, the cell would expand.
That's a very good point. No, we keep them conservative. So the area is conserved. It's a conserved field, and that comes from the conservative dynamics that we're having. So the area is always preserved. Yeah. So if they're space filling and they're incompressible, you can't generate segregation. Yeah, by default. Okay. Right, so we have this pair separation to look a little bit of the mixing and the chaotic type of flow that we have. And you see that it's very much sensitive to activity, as we would expect, and they separate further away when we increase activity. Okay, you say, well, so what? All right, so we will try to do a data collapse, okay? So what controls this fluidization is, as I said, this junction remodeling, this T1 transition. So what we've done here, we plot it, instead of plotting it as a function of time, we plot it as a function of the cumulative the total number of T1 transition up to that time. Okay, so the cumulative T1 transition. So you see that the mean square displacements do not data collapse. There is something there. It doesn't. So there's no relationship between one and the other. On the other hand, there's a beautiful data collapse of the pair separation with the cumulative T1 transitions. Yeah, for all the activities, they always go into a straight line. Okay, so of course it depends on the activity, but the underlying thing that activity does is to increase the frequency of the T1 transitions, right? But the scaling is actually linear. And there's lots of parameters in that model. And the student that was working on that, he spent a lot of time tweaking all of these parameters. And regardless of it, we always see this, this linear scaling. So that was very nice, which tells us that the fluidization is really due to remodeling of this junction. You're changing the junction with the neighbors. And that's very nice. And the final part, I think I can maybe skip, but there's something that where we looked here, what happens if you have a junction here, and what's the probability of having another junction somewhere else? Huh? And how will that affect the motion? There are different kinds of things. So either you gain a neighbor, or you lose a neighbor, and you have all of this combination. What happens in having two successive junctions? And the point is that the pattern where you lose a neighbor and you gain a neighbor tends to happen when the T1 transition are in opposite directions. And you see that when we looked at the, again, this is doing something on its own. These are the two patterns, gaining and losing. And you see that the likelihood of having another junction at the pi is much higher, which is at the opposite edge. Which means that you, this is the crawl, this is the swimmer, pulling or pushing. This is basically the dipolar type of motion. So they have to either push into a neighbor and create a new junction, but then it closes in the back, or the other way, it pulls. So this is very nice. It connects to the coarse graining prediction that we have from a completely different model. Yeah, this is something else. And I think now I can basically summarize the points that the planar cell polarity seems to play a very important role in migration and the fluidization, and that's something that, to our knowledge, hasn't really been explored very much. Fluidization is very important for morphological changes and generates this spontaneous tube formation. It leads to the primary elongation in there for the gastrulates. And junction remodeling is another type of fluidization mechanism that leads to quadruple flows. And that the relative migration and the mixing is controlled by this rate of junction remodeling. And with that, I can thank the principal actors in this group. So Richard Ho is a postdoc who has been doing all the simulations of the polarized particle model, I think. Harish Jain was a PhD student, graduated a couple of months ago, and he was in charge of the simulation with the multi-phase field model. And he previously was a master student with Alex Voigt from Dresden, and he's one of the one that design and kind of have this platform of multi-phase field models. So we sort of inherited that when Harish came over, and it was natural to have the collaboration. And Stig Uwe is the cell biologist colleague that is helping me kind of bridge all these models to actual data and real tissues. So with that, thank you.
Thank you, Luisa. So now, questions? Well, I have had many questions, but more.
Mm-hmm.
Thank you. Very interesting talk. I guess all goes back to the potential, the effective potential you put at the beginning. No, is what explains all this. No, and you say they was purely phenomenological, but there are any hints or why? So why is the cell-cell interaction like that? So let's say, is there any biological ideas how two cells want to align the polarity, or how does that work? Which is the source of that interaction? It has to do with the dynamics of the pressure at the surface. How does that come about?
This is a very, very interesting question. Yes, I know that there was some recent work where people have actually tried to do the diffusion reaction type of equation, because you have these molecules that are polarizing, and you can have the concentration of these molecules and see how they move in space. And in the typical model that they do in that case, so it's much more biologically informed, that you actually have these proteins rather than these vectors. And they derive this effective diffusion reaction equations that look kind of similar to the ones that I have from the co-screen model. But directly to your point, I don't think we have like a one-on-one map, other than it's kind of in the Landau spirit. You know, we have some quadratic interaction. We want something to be in a plane. What define a plane? We have these vectors. What define the surface? We have this vector. So in that sense, it's a very primitive geometric model that seems to reproduce quite a bit. Now, I think when we dig into the biology, we're going to get like confused with a lot of things that matters, right? So
That's correct, yeah.
Yeah.
Thank you.
More questions?
Thanks for the talk. I mean, it was very interesting. For the first model, that you have the development of the axis, can you go farther and put it in a domain that is bounded like a neck, for example, and see how it because when you have the development, first it elongates, but it develops like makes like a curve because it's constrained by the egg.
Yes.
Can you?
Yes, we did simulations
that?
on that. So you can definitely couple it with the confining potential. This is a potential model, so you can add the confining potential, and the particles will feel it, and they will. But in fact, we saw this banana shape type of things without confinement, just by tuning the model parameters. They can also develop all kinds of. It's a very rich dynamics. So I don't know if that answers the question. Yes.
I think so.
Yeah.
But it's interesting to know also that without the potential,
It's still curved.
you still can
Yeah.
see.
Yeah.
Yeah, in the development of the tubes, they grow these vertical tubes. You say in your model, this is created by you impose a polarization in the form of a vortex, then this induces a flow towards the center. But in reality, it has this polarization measured and seen that it's really a vortex. Is this
We imprint it.
you imprinted in your model? What about the biological tubes?
Yes, this, in reality, yes. What keeps the polarity stable in that pattern? What imprints it?
Or if
Yes.
more before that, has this vortex polarity observed when there is a tube formation?
No. To our knowledge, no, not yet. But the fact that it hasn't been observed could very likely be because people have not looked with this type of thing. Like there hasn't been done an experiment where you can probe. This recent paper that I mentioned, where they actually write the diffusion reaction equations associated with the distribution of this PCP proteins, they do see vortex structures. So it seems like even when you have a model that is more biologically informed, biochemically informed, let's just say, you do see this these vortical structures. But I don't know of any sort of observation. But with my biology colleague, that's something that we are planning to look into a little bit more. Yes.
I've been somehow surprised by your comment on which, well, in the first model in which there is no explicit self-motion or activity, and you get some kind of velocity which looks like the active matter. But in fact, if I understand properly, is this something like a normal matter with a magnetic field in which you have forces which are not
in some way.
not with the field, what perpendicular, and you can have continuous looping without changing the magnetic field, for example. Is something similar, or?
Perhaps
But-
that's something I didn't look at. I think I made maybe I emphasized the contrast a little bit more just because this active stress has been derived in the context of active pneumatics from self-propulsion. And then there is this sort of like, oh yeah, that's where it comes from. And what we see here is that you can
Without
also
the-
get it without the self-propulsion. They don't have to be, they can be one without the other. There are two different types of activity sources, one at the level of the stress and one at the level of crawling, the self-propulsion. And there's a link between them, and that has been exhausted, like people have looked at that link between them, but they can also be independent, and we see them independently here. That's what I'm saying. Yeah.
Any other comment or question from the audience, from the people in Zoom? Nothing? Okay. Then thank you, Luis, again.